What safety tests and efficacy studies do we perform in our labs? Once we have established a colony of gene-drive mosquitoes, we perform studies in our laboratories and insectaries to ensure our technology is safe and effective. We first study the mosquitoes in small cages. This allows us to assess their ability to compete with wild mosquitoes and check that the genetic modification is working as expected. If the results in the laboratory colony show the potential for gene drive to spread, we proceed with additional testing, including in large indoor cages that mimic the conditions of a natural environment. At our partner institution, Polo GGB in Italy, we conduct experiments in both small and large cages. In the lab, we cross the gene-drive mosquitoes with wild ones of the same species from different African locations to ensure they genetically resemble the mosquitoes they would encounter in the wild in Africa if released.
In Italy and the US, we conduct studies to evaluate any potential risks and confirm that the genetic modification functions as intended. These studies also help us evaluate how long it takes for the modification to spread and to check whether resistance to the gene drive arises. We use mathematical modeling to predict potential spread and impact of the gene drive mosquitoes on the wild mosquito population if they were to be released in Africa.
Key safety studies are carried out, allowing us to verify that the gene-drive mosquitoes do not pose greater risk to human health, the environment and animals than wild mosquitoes. In Italy and in the United States, we analyze different parameters, such as how long the gene-drive mosquitoes live, their biting behavior, the ability to transmit diseases, and their resistance to insecticides in comparison to wild-type mosquitoes. We also evaluate the mosquito's tolerance to different environmental conditions.
If all safety and efficacy studies are concluded with satisfying results, and if our partners are ready, we will seek permission from the relevant regulatory authorities and agreement from affected communities to import the gene-drive mosquitoes to our laboratories in Africa. The African teams would gather valuable information on the potential field sites in preparation for potential field releases. Together, we can end malaria.